In this video, we're going to discuss the biochemical mechanisms of LTP, or since a lot of people don't want you to call it a mechanism, basically what's happening at the synapse during long-term potentiation. And there is a handout in the module that you should either watch the video and then try to answer the questions on your own, or answer the questions as you're watching the video so that you can use it as a study guide. It's nothing to be turned in, but it's something to help you be more successful on the exam. The first thing that's going to happen is there has to be an action potential. So we have an action potential that comes down the presynaptic neuron here, and that causes release of neurotransmitter into the synaptic cleft. It's going to bind receptors on this postsynaptic cell. Please, I apologize for my poor drawing. I couldn't find a diagram that did what I wanted to do. And so when this glutamate binds these AMPA receptors, that's going to cause sodium, or allow, I should say, sodium to come into the cell. That sodium is going to depolarize the cell. But that's not going to be enough to cause LTP. So it's depolarizing the cell because sodium enters the postsynaptic cell. And again, remember, these are ionotropic receptors. So does one action potential trigger enough glutamate to be released so that LTP happens? No. You need repeated stimulation of the AMPA receptors. And this is because, remember, there's two types of glutamate receptors involved in LTP. We have AMPA receptors and NMDA receptors. And that NMDA receptor has what's called a magnesium block. And so this postsynaptic cell has to get depolarized enough that that magnesium block is released. So you need repeated stimulation of the AMPA receptors, so there is a much higher depolarization of the postsynaptic cell for that magnesium block to be released. So what happens then after repeated AMPA receptor stimulation? So we have multiple axons stimulating the dendrite at the same time, or repeated stimulation by the same axon. Remember that temporal versus spatial summation? When that happens and sodium continues to enter the postsynaptic cell, depolarizing it more, again, we said that magnesium block is going to be removed, as you see here. And this NMDA receptor is a calcium channel. So when the magnesium block at the NMDA receptor is removed, what do you think is going to happen? If this is an ionotropic channel and it's a calcium channel, that magnesium block is gone now because the postsynaptic cell has been depolarized enough. Calcium is going to enter that postsynaptic cell. So we've seen some videos already, or at least they're posted in the module, of what happens in the postsynaptic cell after calcium enters. And so this diagram is kind of showing you some of that here. We have calcium that's entering. It's going to activate an enzyme called CAM kinase 2. So it activates this enzyme. All you need to know is CAMK2, but it's calcium modulate kinase 2. I believe that's the correct name. And it's going to activate that enzyme, and it's going to cause a whole bunch of downstream events that we're really not going to go into just because this is, again, an introduction to a psychobiology class. But ultimately, CAMK2 is going to cause a release of Krebs. And after this Krebs is released, it's going to cause a whole bunch of long-term effects in the postsynaptic cell. One being gene expression being altered. So again, gene expression being altered means more AMPA receptors can be made. This can be for months or years. Again, it's a long-term potentiation. And this is what we think accounts for long-term memory. So let's go over those postsynaptic changes. So we know that more AMPA receptors are going to be built or moved. So there's going to be more AMPA receptors inserted into that postsynaptic membrane, meaning there's more AMPA receptors to respond to that sodium that receptor that's released, that glutamate. That means more sodium can enter the postsynaptic cell. That's going to also result in dendritic branching. So in order to have more AMPA receptors, we need to have a bigger surface area so that there's going to be dendritic branching, and that surface area of the dendrite is going to increase. This results in more synapses with the same neuron. So again, allowing that postsynaptic cell to be potentiated. AMPA receptors also become more responsive. This has to do something with phosphorylation, I believe. And we also said there was genes that were activated, proteins that were made. This is also going to include more NMDA receptors being made and inserted into that postsynaptic membrane. So you have the two biggest changes are more AMPA receptors and more NMDA receptors. As a result, you need to have a larger surface area for the dendrites, so there's dendritic branching. All in all, this is going to increase the responsiveness of the postsynaptic neuron. And so this is just kind of showing you that here. This is a normal neuron. Again, this is potentiated. We've seen this diagram before, but you can see here, it's not specifying AMPA versus NMDA, but you can see here visually that there is more receptors in that postsynaptic neuron. So there's more receptors to respond to the glutamate. And we're going to see there's also some presynaptic changes as well. So the presynaptic changes is that there are going to be some retrograde transmitters that travel back to the presynaptic cell and cause the following changes that also result in this increased potentiation. First of all, you're going to have a decrease in the active potential threshold. So the threshold of that presynaptic cell is going to decrease, meaning it's going to be more likely to fire an action potential. You're going to have an increase in neurotransmitter release because like what you see in sensitization, right? There's more neurotransmitter that's released presynaptically. So that means more neurotransmitter to bind those receptors on the postsynaptic cell. Expansion of axons. So we're going to see collateral sprouting. We saw that in one of our diagrams as well. And we may even see transmitter release from additional sites. All in all, this is going to increase the activity in the presynaptic neuron. So both your presynaptic activity is increased, your postsynaptic cell is potentiated, and that is what we think results in our long-term memories. And this process that we're talking about, these changes pre and postsynaptically, have been studied for the most part within the hippocampus. And this diagram is just showing a little bit of that presynaptic versus postsynaptic, and some of that, you can see how that presynaptically there's changes as well as postsynaptically. But question is that is LTP important for all learning and storage of memory? Gotta think back. Was the hippocampus the location for storage of all memories, storage and retrieval of all different types of long-term memory? No. Especially for episodic memories, as time passes, the cerebral cortex is more important. And again, whether they slowly move into the cerebral cortex or they're both stored there at the same time, it just takes longer for that cerebral cortex to encode those memories. Research, time will tell. And then we also talked about other areas like the basal ganglia for procedural memory, the anterior temporal lobe for semantic memories. So it's important to keep that in mind, that this may not be the only thing that's going on in the brain that allows memories to be stored.